Okay, I think we can start now. People are still joining, maybe a couple of more minutes. Okay, we can start the class now. Hello everyone, my name is Sumati. I'm from University of Hyderabad and I will be taking one of the weekly live sessions for Cell Culture Technologies course from NPTL. The course instructor is Professor Manak Das from IIT Kanpur. So, firstly, uh, what the course entails for this week is introduction of cell culture technology, philosophy and complexity in cell culture, to grow the cells outside the body, cell cycle concept, and dividing cells. So first, we should know a few terminologies before getting into the process. So I'll just give a basic introduction for 4 or 5 slides. I think most of it was covered by the previous students as well in the previous live sessions, but still, if anybody did not attend that. Okay, so Uh, these are few of the terminologies one must know before entering into cell culture or learning cell culture as a technique. So, the first is adherent cells, that is, cells that are able to adhere or stick to the basal surface, either a matrix or even just a cell culture dish that can be treated or not treated. It's called TC treated. And the ones which do not adhere are called non-adherent cells, and then there are other cells which are semi-adherent cells. They adhere at particular conditions, at either at higher confluency or with many media changes or different ways. So few of the cells detach. Those are semi-adherent. The next is attachment efficiency, the percentage of the cells that actually adhere to the vessels of the surface within a period of time after inoculation or after seeding. So that is what I was talking about in the semi-adherent. So there usually the percentage of cells which adhere to the culture vessel also comes into account. Next is aseptic techniques, which we all know. So it is to maintain the uh, cell culture products and the cell culture equipment, instruments, and the cell culture room free of contamination, that is sterile conditions, free of bacteria, virus, mycoplasma, which will affect the experiments if they are also growing along with your cells, so they will give a different result and there will be problem in reproducibility. So cell culture, that is nothing but maintenance of cells in vitro, that is outside of their original body, whether human or animal, everything outside the body is in vitro. Cell line, the result of first subculture of a primary cell line. So basically, all the mammalian cell lines have been isolated from a specific organism, whether it is mice or human, depending on the cell line. So, whatever cells that have been isolated and grown for the first time on the plates are the primary cell lines. And then, once you start subculturing, it becomes your subculture, which is called also called as passages. And then, from that, if you develop a cell line, that is from these cells now they can grow individually without the help of the organism, where you transfect them with SV40 gene or anything which will make them have the capacity of proliferation, whether they are inherently proliferating cells or not. If they have the capacity of proliferation and then you develop such cell lines, they are called cell lines with which you can uh, again inoculate or revive the stock which has been frozen instead of going to an animal again to isolate for primary cells. The second is anybody can ask any question if they want in the middle. I'm okay with it. So next is cell strain. Derived from primary cell culture or cell line, a cell strain has cells of specific type with unique properties of markers. That is different strain of cells with particular properties like HEK or different other podocyte cells or some muscle cell line, colon cell line, something which is unique to those cells. Then is cryopreservation, which I said, like freezing the cells. So the storage of the cells. So whatever cells you grow, you cannot grow them after a particular passage. For every cell line, there is a range of passages. Many people are joining, just excuse me. So maybe I'll stop admitting students after half an hour because it's interrupting the class. Okay. So as I was saying, cryopreservation, that is storage of cells, tissues, embryos, or seeds in extremely cold temperatures. So even preserving seeds for plant culture is also cryopreservation if you do it below a temperature of minus 130 degrees. So this is generally done in liquid nitrogen where you preserve these cells with glycerol and DMSO as cryoprotectant. So this will help your cells retain their capacity to be revived again into a fresh passage. Okay. So next is epithelial cells, cells that grow very close together in order to form continuous mosaic-like sheets. When other cells adhere to each other, they are referred to as epithelial-like cells. So the cells may be exactly epithelial in origin or not exactly epithelial, but if they adhere to each other, they are also known as epithelial-like cells. Fibroblast cells, spindle irregular, responsible for formation of fibers. Everybody knows this. So, finite cell culture, a culture that is capable of only limited number of subculturing rounds, after which the cells cease to proliferate. So, as I was saying, there is a particular number of passages which you do for every cell line. So, if in this case, if it is finite, that means after a particular number of rounds, the cells start dying and then you cannot further subculture them. Most of the cells will survive many passages, but then they also start to lose their morphology and their innate nature which they have, and then they will not give you correct results or proper results when you experiment with them. Then immortalization, attaining a cell culture that is able to proliferate continuously. As I said, like if you transfect it with SV40 gene or some other gene which helps it to attain a capacity to proliferate further
Hello,
into,
ma'am.
yes, yes, please tell me who is this? Who
Ma'am,
was
you are not
that?
audible, ma'am.
Oh, I'm not audible since. Oh, I'm so sorry. Since how long am I not audible?
From starting, ma'am.
Oh, you should have told me. Okay,
Yes,
okay,
ma'am, I have
let's.
messaged you. Yes.
Oh, I didn't see that. I was in presentation mode. Okay, I'm really sorry about this. Let's start once here. I'll
Okay.
quickly go through this. I'm audible right now, right?
Yes, ma'am.
Okay. So, what I was saying till now was cell culture. Before you learn it, we have to know the terminologies. So, to understand various processes and various types of cells and the type of cells you grow, how you grow them, how you want them to be in a healthy condition. So, these are the things which are mandatory before you start your cell culture. So if there is any issue like I'm not audible or anything, please let me know through your mic because I won't be looking at the chats right now. So okay, let's go first, which is adherent cells. Adherent cells are cells that are able to adhere to the culture vessel, that is the Petri plate or the dish, cell culture dish, with the use of an extracellular matrix or even directly to a coated plate. It's also called as tissue coated. One second. It's also called as tissue coated TC treated plates. So here you can see the cells that adhere to the plate. So these are called as adherent cells. Now there are also cells which do not adhere. They are called as suspension cells. And then there are semi-adherent cells wherein a few number of cells will adhere to the surface, and then depending on the culture conditions, the basement that they are adhered to, and even small things like media changes will detach them from their surface. So those are semi-adherent. Next is attachment efficiency. So, like I said in semi-adherent, so here this percentage of cells that actually adhere to the culture vessel plays a very important role after you seed or inoculate the dish. So aseptic condition, we have to maintain aseptic conditions and aseptic uh, equipments. Even the room, the cell culture room should be sterile so that your cells, whatever you're growing, are free of contamination. Because once your cells get contaminated with bacteria, virus, or mycoplasma, you cannot get efficiency in your results, and there will be very less proliferation, sorry, reproducibility of your results. So once if you think you're getting a good result, but the next time you did everything same but you did not get the same result, that could be due to contamination which is affecting your treatment or experimental conditions. So aseptic conditions are very important. And then next is cell culture, that is nothing but maintaining the cells outside of the body of the original organism. And then cell line, that is the result of first subculture of primary cells. So when you isolate a cell from an organism and then grow it for the first time on a culture dish, it is called as primary cell line. And then when you immortalize them and subculture them into different passages, it's called as passage 1, passage 2, etc. So then it is called as uh, subculturing of cell, wherein it becomes a cell line. So a cell line is immortalized cells which have their individual capacity to proliferate. Now they do not require an organism or a body for the next time when you want to seed them or grow them. Cell strain is derived from a primary culture cell line. The cell strain has cells for a specific type of unique properties or markers. So Am I audible? Am I connected in the meeting?
Yes, now your voice is coming, ma'am. Yeah, ma'am.
Okay. So it was a power cut situation. Let's continue. So, oh, people are still joining.
Excuse me, ma'am.
Yeah.
Ma'am, how do they immortalize the cells?
So the you transfect the cells with proliferating genes like SV40 gene. Have you heard of SV40? So most of the times we use such kind
Yes,
of
yes.
genes
Yeah.
which give the cell the property for proliferation. So that's how you transfect your cells, your primary cells with this, and then you subculture it slowly, and then the cell attains the capacity of proliferation. Okay.
So, ma'am, do they loses some properties like any of the important properties from the primary cell line?
So it depends because most of the in vitro cell lines are not exact replica of what they do in your body, because in your body they also have connections and signals that they get from different cells other than themselves. So once you isolate from the body, they are obviously going to be a little different than how they behave in your body, but more or less you will get a similarity which you can use first in vitro. So then if you want to see how if whatever functioning it is having in vitro conditions, if it is true within the body also, then you have to go to in vivo studies. So when you transfect a cell, it depends again. If it's a completely differentiated cell, it will definitely lose its properties, differentiated properties. So that time what you do is you have permissive and non-permissive temperatures for these genes. So when you want to proliferate them, you will transfect after the transfection and cell line has been established. It will be known that at 33 degrees temperature, if you maintain the cells, they will proliferate. So that time they will not have the properties of the original cell. But then when you shift them to 37, so in 33 the SV40 gene will be active. So in 37, when you shift them after you know that you have a required amount of cells, if you shift them to 37, then this gene will be silenced, and then it will start to differentiate and it will regain the original properties of the primary cell. Hope I answered your question.
Yes, ma'am. Yes, ma'am. Thank you.
Okay, so let's continue.
Ma'am, now I'm having one doubt.
Yeah, tell me.
Ma'am, if we again shift the cell to the 33 degrees Celsius, will it be again going to be like proliferating again, or else like it will going to be de-differentiate, like it will lose the characteristics of the primary cell again while dividing, again while proliferating. So
Again,
is
that
that we can shift the cell
depends.
again to the 33?
Usually that is not advisable. Like you said, it's a very good doubt. So, like you said, it might de-differentiate, it might, lose its capacity to proliferate. Some cells might retain the capacity and then start to proliferate, but you cannot be 100% sure. You can never be 100% sure, but you won't be even like 95% sure that all the cells are doing this. So, usually that kind of process is not done. Once you differentiate them, you have to use them for your experiments. Then you will get definitely some problems if you again put them back to 33 and try to proliferate them. Instead of that, you can go with a fresh stock, again seed them, revive them, and then start the process. Okay.
Okay, ma'am. Thank you, ma'am.
Okay, good. So let's continue quickly. Where was I? Okay, so now cryopreservation. So, sorry, cell strain, right? So, cell strain is a primary cell culture or cell line, a strain that has specific type of unique properties or markers that are specific to a particular cell. Like if you want a cell from say colon. So you have colon cell lines. So these are particularly having characteristics of colon cells only. You cannot again make some other cell with this. And then you have cells for kidneys. You have podocytes, which are kidney cells. So they have completely differentiated cells. So they have those particular. So these are called as cell strains. Now cryopreservation. So once you start subculturing and then you go to number of passages, and then after a certain number of passages, what happens is the cell starts losing its healthy condition. It loses its morphology, some of the functions. Most of the cells will be in senescent stage, they'll be dying. So there is always a particular number of passage numbers which has to be done for a cell line till which it will be healthy and it will be useful for your experiments. So after this, you cannot keep continuing and keep on subculturing the cells. So it will damage the cells. So what you do, you go to a particular number, like say 20 subcultures or 30 subcultures, depending on the cells. And after that, you again go back and revive a new vial, new stock. So how you get the new stock? When you initially passage it, your first, second subculture itself, there you have to freeze some of the cells. So you use glycerol and DMSO, which is a cryoprotectant. This will help your as a stock for you for the next set of cells. So usually this is done in temperatures below minus 130, that is usually in liquid nitrogen, which will help maintain the functional uh, functionality and morphology and all the conditions of the cells without damaging them. So this is called as cryopreservation, which is very important. So once you get a cell, you can cryopreserve them and then you can go on with the same cells for years together. You will not have to get another cells unless there is some problem. Next term is epithelial cells, cells that grow very close together. So in body generally, you know, epithelial layer is the topmost layer lining to most of the organs, or outside the body, the skin. All these come under epithelial cells. So when the unique property of these cells is they adhere to each other. So there are some cells which are not exactly epithelial in origin, but even though they have the capacity to adhere with each other, so they are called as epithelial-like cells. Next is fibroblast cells. These are very commonly used spindle irregular shaped cells which helps in formation of fibers, and it is hard to distinguish fibroblasts from other cells in culture, leading to the cells termed as fibroblast like cells. This is a different thing. So finite cell culture, like I was saying, the number of passages is limited for the cell lines. So a cell culture is capable of only a limited number of subculturing rounds, after which the cells cease to proliferate. Immortalization, that is attaining a cell culture that is able to proliferate continuously. Like I previously said, when you transfect the cells with SV40 gene or something similar to it, you will make the cell gain the capacity to proliferate on its own. So that is known as immortalization. Then passage, like I said, after every subculture, it is called as a passage. So the transfer of cells from one culture vessel to another. A more specific term is subculturing, where the cells are first subdivided before being transferred into multiple culture vessels. A passage number will refer specifically to how many times a cell line has been subcultured. So the more uh, specific term or technical term is subculturing. Passage is how we term them in the lab, so that you know how many number it has been, passaged from how many different culture vessels. So when you take and you revive your cells and then you start passaging, it is P1. Then next subculture is P2, next subculture is P3. So like that you can go till P10, P20, P30 depending on the cell type. This is to regulate the number of subculture you do to maintain your reproducibility. Next is primary culture, a culture started from cells, tissues, or organs that are taken directly from an organism. Suspension culture, a type of culture where cells multiply while suspended in their liquid medium, like cells which do not have the uh, ability to adhere, like blood cells, erythrocytes, lymphocytes, all these cells are suspension cells. So you grow them in flasks, so they are suspended in the media. Next. So let's go into a little technicality of the procedures. So first, how you start your culture is you have a stock. So from the stock, you take the vial, you thaw the cryo vial in 37 degrees water bath. So this will help in decreasing the damage to the cells. So when you thaw it in 37, they will immediately come to the healthy condition. So then you transfer this contents into a conical tube, and that even the media which you are using from now on, everything should be in 37. So what you do, you either leave your media in room temperature. If it's like a tropical country like us, that is more than enough. So it will automatically come to some 35 degrees centigrades. If not, if you are in a very well AC lab, or if you're in a different country with lower room temperature, there what you can do is you have to pre-warm them in the same 37 water bath. So then your cells will not get any stress due to temperature change. So after transferring, you centrifuge this cells for 200 G, or even 1000 RPM is also fine. So you centrifuge them for 3 to 4 minutes, and then you discard the supernatant. You resuspend the pellet in fresh media, and then you again inoculate or seed them into culture dishes which already have media. So depending on the size of the culture dishes, the volume of the media will change. Now, after this, this is done. This is your day one. So after this, the cells have grown, they have proliferated. Now you want to go for the second passage, that is subculture then. So what you do, you have to, if there are adherent cells, what you have to do, you have to detach them. So for that, you have to use some enzymes like trypsin EDTA. So here, you remove the culture medium, you add 1 or 2 ml of trypsin EDTA, leave it in 37 degrees or room temperature for 3 to 4 minutes, and then you have to neutralize the EDTA, or neutralize the trypsin, sorry. So what trypsin does is trypsin is an enzyme, so it cleaves the attachments of the cell to the surface. So if you don't neutralize it, it might start cleaving the cell itself, and then it will cause a problem. So you have to neutralize it with fresh media, and then again collect it in a conical tube, centrifuge it, same, resuspend the pellet, and then again freshly inoculate into a fresh culture dish with fresh medium. So this will continue with several subculture steps. Then in the first or second passage, usually we freeze one of the plates. So even that time you trypsinize, you collect the pellet. So now instead of resuspending it in fresh media, what you do is you suspend it in freezing media. Freezing media usually contains your culture media with DMSO. So DMSO helps in cryopreservation. So that's how you freeze your vials, and then you freeze them in minus 130, that is in liquid nitrogen, and store it in either liquid nitrogen tank, or if they are not very sensitive cells, you can also store them in minus 80 freezers. But liquid nitrogen is more preferred. So these are the cell culture dishes. So you can see different sizes, different volumes. So the top one with a blue cap, it's called T25 flasks. So based on the size also, it has again T25, T175, T75, etc. Depends on how much cells you want, you will choose your culture dish. Then the uh, dish that is adjacent to that, this one is the 60 mm culture dish, and this is the 100 mm culture dish. This is a 24 well plate, this is a 6 well plate, this is a 96 well plate. All of them can be used for different experiments. If you need more cells, like for
Excuse me, ma'am.
yes.
Ma'am, I had a doubt. The which property of the cell determines the number of subculture that a cell line can undergo?
It is not a particular property as such, but it 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 depends on the proliferative capacity of the cell, if I'm not wrong. I'm not really sure about that property, but it's different things. It's like few cells have that sturdiness to, survive outside the body in these kind of artificial conditions, but few cells do not. We do not exactly know why it is, but with practice and with experience, people have given particular range of passages for every cell line. I'm sorry, I don't know the exact reason if there is, but as far as I know, this is what we practice.
Ma'am, thank you.
Okay.
Ma'am, I want to know about the EDTA and trypsin wala thing. Like I was not able to concentrate on the words you told, as it was why the voice was breaking. So the trypsin EDTA, what is that thing, and why we have to add all those things in that subculturing process?
Yeah, sure. I'll repeat that for you. So, when we first inoculate the cells into the culture dish, what we do is attach them to the plate. That is, if they are semi-adherent or adherent, mostly, either a lysine layer or an ECM layer, a collagen layer, or just a TC treated plate is used, the cells go and attach to the base. They need a base to grow, especially for adherent cells. If it's a suspension, trypsinization is not required. These are only for adherent cells and semi-adherent cells. So now when you want to subculture, like this is your first inoculated plate. So from this plate, you have to now subculture into your second plate. How will you take your cells out without damaging them? So for that reason, we use enzymes like trypsin. So trypsin is combined with EDTA to give get activated at 37 degrees, and it will act on the proteins which help in attachment of the cells to the basement. That is anything that the cell attaches to. So what it does, it will break the connection between the cell and its adherent medium. So if the cell are adherent and adhering to each other, it will also break the cell connections as well. So that is the reason we have to also neutralize this after incubating it with trypsin. So when you neutralize it, the trypsin action will be inhibited. It will get inactivated. So that time you can so this is also important for your cells to detach from each other, because the next time you inoculate, if your cells are clumped, so they will grow on one another. Okay, you get it? They'll grow on top of each other, and every cell will not grow very efficiently. So the basic cell culture thing that we all have to do is grow a mono layer of cells. That is, in the entire plate, only a single layer of cells have to be there. The cells should not overlap on each other and not inhibit the growth of other cells. So there also the trypsin helps from breaking the cell to cell connections and give you single cells for the seeding for the next subculture. Okay. Hope your doubt is cleared.
Yes, ma'am. So the EDTA will play in the neutralization of the trypsin, I guess.
No, no.
Oh,
Neutralization is done by a fresh media. If you like, the media has serum. So the serum, the components will help in neutralization of trypsin. EDTA trypsin is to maintain the trypsin without degradation and help in activity of trypsin in the media.
okay, ma'am. Thank you, ma'am.
Yeah, sure. Uh, let's go to the next slide. Okay. So as I was saying, these are the different cell culture dishes, and then this is the laminar airflow, or it's also called as hood in general lab language. So here we this also has to be aseptic, sterile, free of contamination. So we wipe it completely with ethanol, 70% alcohol, either isopropanol or ethanol. And then we also have a UV lamp inside the airflow. So we turn on the UV lamp before using it for 15 minutes and after using it for 15 minutes to keep it free of contamination. And then this is your CO2 incubator. You have a glass door for it, and then you have a main door outside that. You have, depending on the size of the incubator, you have different number of racks like this where you can place your dishes, and at the lowermost rack you will have to place water to maintain humidity, otherwise the media will get evaporated because of the temperature. And then it's a CO2 incubator, so you have to have a supply of CO2 from your gas cylinder, which will help in maintaining the pH of the media. Okay, so this is a basic requirement for a cell culture lab. Next is there are different types of cell culture models that you can use. One is primary cell culture, like we discussed, then it goes to a long term. Sometimes when you can't uh, make a cell line, if you can't make certain types of cells into a cell line, you always isolate it again from the organism and grow it in the in vitro. So they always are done as a primary culture line only. Most of the cells which are converted as a cell line are for long-term use. You do not need an organism again there. So then there are again 2D co-cultures, 3D co-cultures. So here you have a different kind of plates which are used, which will help in s- uh, which will help in culturing more types of cells. If you want to see the complexities of connections and signaling between different types of cells. And then there are spheroid cell cultures if you want to see the effects at a tissue level, not just as a cell level, because cells we usually grow in monolayer, as we said. If you want to see at tissue level or organoid level, recently have come very useful as a model. So there you have again different types of equipments and different types of culture dishes that are required for that. Okay, so till now, this is what I have for you to offer in the introduction and in the process. So after this, I will be going for the assignment. So if you have any doubts still here, you can please ask me. Then we will head to the week one assignment. Okay, shall we go ahead then? So here also I'll be telling you a little bit of different things. Okay, so so I think you have already done, most of you I hope have already done first or second live session. So if you have done, you can answer the questions. So it will be an interactive round. So let's start with the first question. So first is, in what phase of cell cycle the cell either progresses towards DNA synthesis and another division cycle, or exits the cell cycle to commit a differentiation? Anyone answer?
G1.
G1.
Yeah. So the answer is G1. So you know the different phases of cell, that is G1 and G2, which are both growth and metabolic, metabolically important for the cell, because that is the phase where the cell grows. And so this is your first G1. Then you go to S phase where your DNA replicates. This is also the synthesis phase. And then you go to G2, and then you undergo cell division, that is mitosis. So here again in mitosis we have prophase, metaphase, anaphase, telophase, cytokinesis. Okay, so the answer where the cell either progresses towards DNA synthesis, that is your stage of the cycle where it is before your DNA replicates. That is what is the first part of the question, that is G1, and another division or cycle, or exists as a exits the cell cycle to commit to differentiation. So there are two routes for the cell at G1 phase. One is it can continue with its proliferative property and go into DNA synthesis and replication, or else if that cell has been selected to stop proliferation and go into its differentiation, then from G1, instead of going to S phase, it will go to G0 phase. G0 phase is also called as quiet or quiescent stage. So now here, so quiet in the sense, proliferative cells are very active cells. So the cell which do not proliferate, they are named as quiescent cells. So the quiescent cell will now differentiate and gain its property to become a specialized cell. So I have given some pictures for real images. light microscope images for these different phases within mitosis, just as a extra information. So this is interphase. This is how the nucleus looks in the interphase. This is how it looks in the prophase, where the chromosome starts to condense, and then there is metaphase where they align at the center. You can see a metaphase, the equator, and then there is anaphase where the spindle fibers are formed, and then they are pulled apart to the different opposite centromeres, centrosomes, and then telophase where now the separation is complete and the karyokinesis is done, that is nuclear division is done, and now the cell will also start to divide, that is cytokinesis after that. Then again it goes to resting phase. After this, the cell will become non-dividing. Next question. Which of these is not a constituent of animal cells? Which is the answer?
Cell wall.
Cell wall.
Cell wall. Yes, correct. So cell wall. So you have to know the difference between cell wall and cell membrane for this. So cell membrane is also known previously as plasma membrane, is found in cells, in all the cells, and it separates the interior of the cell from the exterior environment. The cell membrane consists of lipid bilayer that is semi-permeable, so it is permeable to certain salts or ions, but not to everything. Cell wall, on the other hand, is a rigid structure that surrounds the cell and provides them structural support, shape, protection, and acts as a protective barrier. So, cell wall is seen in plant cells and other cells, maybe in fungus and others. And depending on the type of bacteria, even in bacteria, so in bacteria or plant or fungus, depending on the type of it, the components of the cell wall also will change. Mostly it's like a polysaccharide layer or lipid polysaccharide layer, there are different things. Cell membrane is seen in all of them and it's mostly a lipid bilayer. Let me see, still few people are. Okay, it would be better, I think, if you all join within the first half an hour of the class because it's interrupting. Where were we? Okay, cell wall. So these are all the components of a cell. Just a little more information on this, like you can see the nucleus, then there is nucleolus, there is chromatin around it, there is smooth endoplasmic reticulum, there is rough endoplasmic reticulum, there are peroxisomes, vacuoles, cytoplasm, Golgi apparatus, Golgi vesicles, and then ribosomes, intermediate filaments, mitochondria, very important, and plasma membrane. Microtubules are also very important for maintenance of the cell shape. Centrosomes, microfilaments, lysosomes, secretory vesicles, etc. So this is how they are depicted in the textbooks. I will show you also a TEM image, that is a transmission electron microscopy image, how the cell actually looks when you magnify it like more than 1 lakh or 5 lakh times. So this is how it looks in real. So here you can see a similar kind of nucleus with nucleolus. So one difference you can see here is here when they depict, they show a nucleus as a single round sphere, but generally it won't be like that. It will be spread out in different places, and then there will be heterochromatin region, euchromatin region, and then there is endoplasmic reticulum. You can see like lines here, and then there is Golgi apparatus. You can see the nuclear membrane very clearly, and then there are mitochondria that are spread across the cell, and then there are vacuoles and other things. And most of these small, much more smaller things like microtubules, ribosomes, and chromatin strands or intermediate filaments, these will not be visible even after such high power magnification. For that, you have to go to further sophisticated uh, process. Okay, the next question. Next is, in what phase of cell cycle DNA replicates? Answer?
S phase. S phase.
S phase, correct. So this is a image of cells which are immunolabeled, fluorescently labeled. So using a cell cycle chromophore, which is attached to pCNA. pCNA is a proliferating cell nuclear antigen. So they are seen more in proliferative cells. So this helps in, distinguishing the nucleus, which is in division state or synthesis state, etc. So here you can see this is the entire cell and this is the nucleus. So this is how the nucleus looks in G2 phase, and then it enters mitosis. What is after G2? Mitosis. So in mitosis you can see the starting. You can't exactly see like the prophase, metaphase, anaphase. They're all not very like distinct separate. It's a process, dynamic process that goes on. So for that you have to tag the chromosomes, which is not done here. This is only pCNA, so you cannot see the chromosomes, but it's happening here. Okay, and then at the end of mitosis, you see cytokinesis, and then there is G1 phase, and after G1 phase you see S phase. So then S phase is also early S phase, mid S phase, and late S phase. So here you can see more fluorescence because the replication is starting, and these tiny dots are nothing but replication foci. That is, at different places the DNA starts to replicate. This is G2 after S phase, and then G2 phase, G2 to mitosis, and then mitosis again to cytokinesis, again G1, again same cycle. So as I said, in mammalian cells, DNA replication takes place at discrete nuclear sites. These are called at replication foci, where newly synthesized DNA accumulate because they start at different points in the nucleus. Okay, next is in RGD, G stands for what? Anyone? Anybody? Okay, this is a tough one. So G stands for glycine. You know the nomenclature, single letter nomenclature for amino acids, right? What does G stand for? What does K stand for? R stand for? So the answer for this is glycine. G stands for glycine. So this is actually RGD means arginyl glycyl aspartic acid or arginine glycyl aspartic acid. So this is a very common motif that is found in the uh, region where the cell tries to adhere to the extracellular matrix or even to other cells. So this is a motif. This is a three amino acid motif, that is arginine, glycine, aspartic acid motif. So why this is important? Because this is, first of all, it is found in all the species starting from Drosophila to humans. That is Drosophila, everybody knows, fruit flies, and then to the most complex humans. And then why this is important is this RGD motif has been used widely to target tumor cells in therapeutics and in drug designing or even in diagnostics as imaging agents. So you can see here in the second image, this RGD peptide is a theranostic application, that is therapy and diagnostics both together. So in therapy, the RGD conjugated drugs are used. So this will help in targeted drug delivery or effective for more for tumor cells, and then whatever then the drug action is there on the tumor cell, it will lead to cell death. And in diagnosis, RGD modified radionuclide is used. Like there is some radioactive tag to it or some fluorescent tag, anything that is visual. So that is attached to the RGD. Then the RGD attaches to the cells. Then this tag is taken up by the cells. So this will give us high contrast image of these cells in diagnosis. Not only this, RGD peptides can also be used as functional polymers to improve the cell adhesion. Like I told, this is your RGD. which attaches with integrins. So, and then on the other side, it attaches with this ECM. So, it helps in adherence of the cell, increasing it. So, it can promote cell adherence to a matrix, or it prevents cell apoptosis or speed up tissue regeneration. So, this is very useful in medical applications. Next question is, what implies that the specialized properties of the cell are lost irreversibly? Which is the answer for this?
Differentiation.
Sorry? Yeah. Please
Option
tell me.
D, ma'am. De-differentiation.
Yes, so it is de-differentiation. So once the cell has de-differentiated, it cannot again go back. So its original property is lost. Now it will become a prior, like a progenitor cell. So this is a picture for you to understand. So once differentiation is done, then it will de-differentiate into a stem cell-like state, that is a progenitor-like state. So then all the properties of this differentiated cell will be lost in the cell. The cell will not have that property. But anyways, this is a transient phase. So what does that mean? So from here, it can again go back to this, or it can go to a redifferentiation. It can become some other cell, other type of cell, and then it can, or if or it can become a proliferative cell. So it can undergo cell cycle now. If in case the cell, due to some uh, stress condition or a condition or a stimuli which is not able to invoke the capacity of the cell to, complete its phase. I told you, like, this is a transient phase, so it has to complete to become something else. So if it does not happen, if it can't go back and become this cell, or if it can't go and become a different cell, or it can't proliferate further, then there is no use of this cell. So then this cell will go to cell death. So this is one of the very major processes in pathology. So this is very important that the cell completes its next steps and become some other cell. So if you want a definition for the above, all the options, cellular differentiation is the process by which a stem cell changes into a more specialized cell with special features, unique properties. During this process, the cell loses its developmental potential and gains specialized functions and phenotypes. Then comes, one second, please join very early. It's 1 hour late. Okay. Then comes cell selection. It is a process that involves sorting of the population of cells with different properties or fates. A cell fate is determined by cell selection. Then cellular adaptation. So here the term given is de-adaptation. First, we'll see what is adaptation. Adaptation is the capacity of cells to answer different sorts of upgrades or negative controls in the ecological changes. So, adaptation is whatever signal the cell gets from its exterior, it adapts to that. So, what is de-adaptation? When you take out a cell from its original place, it will be where it was getting signals from different other cells, and because of those extra signals, it was functioning in a different, in a specific way. But now you have picked that one particular type of cell and put it in a different environment where you don't have all those signals which it was getting before. So now it will de-adapt because it has to survive. So it will de-adapt itself and it will lose few of its previous properties. Now, next is de-differentiation. That is what we saw now. It's a cellular process that causes a differentiated cell to revert to a less differentiated state, sometimes even back to the stem cell-like state. De-differentiation can be a response to a stimulus or can be part of trans-differentiation, or where a cell reverts to a less specialized state before creating new cells. So whatever I explained here, everything is de-differentiation. Next, cardiac cells are mostly characterized as what type? What type of cells are cardiac cells? Anyone? What are they?
Muscle
Are they
cells.
bone? So they are muscle cells. So because they have the property of beating, right? So they contract and then they expand, so they are muscle cells. So here I wanted to show you how the cells look when you stain them. Usually, how we see a tissue is we uh, have very thin slices of them, thin sections like transverse sections or longitudinal sections, and then we paraffinize them onto the slides like a very thin layer so that the light passes through it and we can see through a single, as much as possible, the most thinnest layer in the microscope. So since they are all transparent, like if there is no blood, if the blood has been profused out before sacrificing the animal, you won't see any color, everything will be transparent. So you need to visualize it. So here we will, so here we will use specific stains like hematoxylin, eosin, stains which stains the nucleus and the cytoplasm, and many other stains are there. You can read about them. So we use different stains to visualize this. So this is how a cardiac muscle looks. This is transverse section, this is longitudinal section. So this, the blue spots that you see are the cell nucleus, and the pink regions that you see is the cell cytoplasm. Then there is it has been outlined here, you can see. Then there is also intramuscular capillaries and all this. This is not, this is very detailed. I just wanted to show you how a cell and a nucleus looks upon staining. So these are called as histological staining. Uh, and then other cells in the options are like bone cells. So these are osteoblasts, osteoclasts, osteoids, different bone cells, how they look. Because it's a bone, it's more of a different nature. You can see that they look more fluid in nature than this. So you should be able to differentiate into different cell types after staining, so that you will know what type of cell there is. So you have to, look into all these types. And this is how a neuron looks after staining. So this is the nucleus, this is the dendrites, this is the glial cells around it, this is the axon. So this is a section of the brain, so where you can see the neuron. And then there is what? There is epidermal cells. So epidermal cells, we have all done the diagram of the skin. So this is the stratum corneum, stratum granulosum, stratum basal. So here also the blue color ones are the nucleus, and then the cells are like very longitudinal. These are epidermal cells. Next question. HeLa cell lines are derived from which cells?
Cervical
Anyone?
cancer cells.
Yes, cervical cancer cells. So different, these are how the cell looks under light microscope. So different cells are there, like strains of cells, like they are derived from a particular type of tissue. So here you can see how the cells look. So when you culture the cells, if they are epithelial in nature, many of them look very similar. But still, if you have experience in them, you can, once you get acclimatized to different types of cells, you practice them using. So you can also, differentiate by just looking at the cell. So this is from ATCC I have taken. So from there you can buy cells if you don't have any organism to isolate or anything, or if you don't have previous stocks, or if it's a new lab. So this is a very good company which supplies very uh, certified cell lines. So this is how the epithelial cell looks. You can see the scale bar. This is 100 microns. So you can see how big is the cell. Okay, so this is confluency. So I can also tell you here about confluency in cell. So usually when you grow cells for an experiment, for particular type of experiments, you need a certain amount of cells. So that is called confluency. Confluency is nothing but the percentage of cells you can see visually. Like here, if you see, the confluency is more than 50% because how much area of the plate the cells have covered. So here you can see it has covered more than 56, about 60%. So the confluency over here is 50 to 60%. But on the right hand side, you can see the confluency is more. That means the percentage of area the cells have covered on the plate compared to the empty spaces is much, much more. So this is around 80% confluency. So depending on what type of experiment you want to do, you will decide your confluency percentage. So there are other examples I just wanted to show, like L6 from rat, C2, C12 from mouse, primary human skeletal muscle cells. These are muscle cell lines, and then there is LAPC4, LNCAP, C42, 22RV1. These are prostate cell lines, and then PC12 is a very well used neuronal cell line. Okay. So next question, which of these gases along with its buffer is used to maintain pH in cell culture medium?
Carbon dioxide.
Yes, carbon dioxide. So this is a small schematic. So this will how your medium look when you leave it in your incubator, even inside the incubator. Outside the incubator, this process is very fast. Inside the incubator, it will take few days, and also it is also a check for contamination. This is very important. So here, what is how carbon dioxide is useful is the you supply usually uh, between 2 3 or 2 to 3 to 10%, anywhere between there you supply the carbon dioxide percentage. Standard everywhere we use is 5% carbon dioxide in the incubators. So the carbon dioxide in the air that is pumped into the incubator will get dissolved into the medium. So once it gets dissolved into the medium, the carbon dioxide and H2O together will form carbonic acid. So carbonic acid helps in maintaining the buffer system. The buffer system is nothing but some kind of bicarbonate ion or sodium bicarbonate. These are generally present in the media which we use. So these are buffering systems. So when the cells grow, they secrete several things. They utilize things from the medium. So then the H plus concentration changes in the medium. So to maintain that, this bicarbonate helps. So this carbon dioxide has to be continuously supplied so that this bicarbonate equilibrium is maintained, and then later few of the bicarbonate will also be served as sodium bicarbonate reserve. So this bicarbonate is only actually present in the medium, which acts as a buffer system. So along with this, we have phenol red. So phenol red uh, is indicator dye, which we use in many chemistry titrations and all. So this will help us. How will we know? We have done all this. Okay, we have supplied carbon dioxide. How will we know that the carbon dioxide is getting pumped? We have sensors for that. But what if the sensor is not working? So how will you know whether the buffering is happening or not? So for that, they also supply the media with this phenol red, which is a pH indicator. So when the carbon dioxide levels are down, and then the acidity of the media increases or the alkalinity of the media increases, either ways, the color will change. The media has to maintain at a neutral pH of 7.8. 7.4 also is manageable. So 7.8 usually it will be there in the outside, and the inside it will maintain between 7.8 to 7.4. So till 7.4 it is healthy for the cells, but once it decreases below 7, then the medium gets very acidic, and then the cells will be damaged due to the acidic nature. And also, so if but this should happen gradually. So you inoculated the media today, you inoculated the cells today, fresh media is there, you put your plate into the incubator, and tomorrow it will go slowly from this 7.8 to 7.4. Usually we change media for every 2 days, so to avoid this change between 7.4 to 7. So if you leave it for another day, it will be somewhere between 7.4 or little more less than 7.4. On the third day, it will become something like this. It will look like this. So that means it's already a little late for your media change, but you have to change immediately. If you change it immediately, everything will be fine again. It's always better to change your media during the state itself, that is within 48 hours. So, and then if you leave it for more days, you forgot about the plate, completely, it's out of your mind. So that time you see that your media will become completely yellow after 4 5 days. So that means now you cannot, even if you replenish your media, your cells will already be damaged. So now you have to discard this plate. And one other, so this will happen gradually with each day. But what if you inoculated your media cells today in fresh media, you put it in the incubator, but then you came next day, you just wanted to check if your cells have attached or if they're looking healthy, if the number of cells you seeded are correct, and you know their morphology is good, and then suddenly you see that this is not there directly from this color which you inoculated your cells in. Now your cells are yellow. That means what? There is contamination. So if there is bacterial contamination or fungal contamination, mostly bacterial contamination, what happens is they start utilizing all the media uh, components, and then they also secrete their own things. So then immediately your media color changes from this kind of fuchsia pink to yellow. That means your plate is contaminated. So immediately you have to put it under the microscope and check it for different types of contamination, or else discard the plate directly. So you should not take the risk of putting a contaminated plate into an incubator and cause risk to other plates around it. Because if you leave it for more days, if it's a sporulating bacteria or fungi, they generally have spores. Once the spores spreads into the incubator, then you have to completely discard all your plates, all your cells, and then completely sterilize your incubator, deautoclave it again, and then only you can use it. You have to fumigate your entire cell culture. Okay, so always better to use a media with phenol red. It will help you in checking, keep a check on both buffer and contamination. Next, so this one, what implies that the synthesis of specific products or other specialized functions are under regulatory control and can be reintroduced or reinduced if the right conditions are provided. What is the answer for this?
De-differentiation.
The answer is differentiation. So the implies that the synthesis of specific products or specialized function are under regulatory control and can be reinduced if the right conditions are provided. Am I correct or am I wrong? So if you, so, like your cell has been proliferating, it has a stem cell like property. Now specific conditions, specific products are being produced and synthesized extra other than usually what it was producing, and now it wants to take up some specialized functions. Okay, and these specialized functions will be controlled by some regulatory mechanisms. That is nothing but differentiation. So once your cell has this new properties, it is differentiated into a cell and it can be reintroduced if the right conditions are provided. That is de-differentiation. So your first part is differentiation. They are saying if reintroduced again, then it can be go back to that proliferative or stem cell like stage. So this process is differentiation. So this differentiated cell can be again de-differentiated back. So the same image which I showed before, this is your differentiation cell. From here, if you go, it's de-differentiation. But what they're talking is something that is before this. From here, it has become a differentiated cell. Okay. So now the next one is, which of these is a non-adherent cell type? Anyone? I think I spoke about this in the beginning, if I'm not wrong. Adherent, non-adherent. Am I audible? Anybody? Am I audible? Somebody please say yes, even if you don't know the answer. I hope I'm audible. I'm not getting any answers.
Yes, ma'am. You are audible.
Okay. So, okay. So, what is this? This is, which of these is a non-adherent cell type? Tell me which of these does not adhere? Bone cells, do they adhere? Hepatocytes are nothing but liver cells, do they adhere? Erythrocytes are RBCs, do they adhere? Neurons are neurons, brain cells, do they adhere? What happened? Okay, so the answer for this is erythrocytes. Erythrocytes are nothing but RBCs, so these are non-adherent cells. The others are all adherent. So these are some of the common morphologies of adherent and suspension cells, suspension or non-adherent. Same thing, suspension cells and non-adherent cells. I wanted to show. So these are adherent cells, that is nothing but they grow attached to a surface and then they spread out, they become flat, and usually on the plates, if you grow a monolayer, it is a two-dimensional arrangement. Suspended cell lines in media. So some of the neuronal cells like NG108 cell lines, these are sorry, these are also adherent cells, neuronal cells. The suspended cell lines are T cells, mostly blood cells like erythrocytes and T cells are all suspension cells. And then there are also monocyte-like cells. These are all suspension cells. So you grow them in flasks, conical flasks. You put them in the media and then you put them in the incubator and then you replenish the media by centrifuging the suspension cells. Nobody asked me this. Why? How will you replenish or change media for suspension cells? How do you do it? You collect all the media, you centrifuge it, spin it down, discard the old media. If you're growing it at a small scale, then again you resuspend the cells and then you add fresh media. If you're growing it at large scales, you have big fermenters or big incubators. So there they have one inlet and one outlet where the old media will be discarded out and then fresh media will be constantly added. Okay. So this is how you change media. Changing media is also very important. It will help your cells be healthy. It will help your cells maintain their specific properties, and it will help them, proliferate quickly. So that is all today. I hope you all understood. If you have any questions, you can ask me. Anyone, any questions? Ibrahim has joined just now, so I don't know. The class almost done. So what I'll do, because we have little bit more time and people are still joining, so I will just quickly give the answers for the assignment. If you all were in some confusion or missed any of the questions. So let's just go back to the assignment. Please join in the first half an hour of the class. Okay, so the first question is, in what phase the cell cycle the cell either progresses towards DNA synthesis and another division of cycle, or exists the cell cycle to come in differentiation? The answer for this is G1 phase. So this is what extra information that I wanted to give you there, that G1 is nothing but growth and metabolic phase, and S is replication phase, synthesis phase. G2 is also growth and meta metabolic phase, and it is also a preparatory phase for mitosis. So mitosis, prophase, chromosomes condense; metaphase, chromosomes align at the center; anaphase, they duplicate, and the duplicated DNA segregates, and then in telophase the chromosomes are decondensed again back, and then cytokinesis, that is cell division, occurs, splitting of the two daughter cells occurs, and then the cell can again
Excuse me,
divide.
ma'am.
Yes.
Ma'am, you are not audible properly.
Okay, am I? 1 second. Okay, am I audible now? Hello, am I audible?
Yes, ma'am.
Yeah.
Yes, ma'am. Yes.
Okay. Okay, we were just going through the questions again. So this is your first answer. G1 phase is where uh, the cell either progresses to the DNA or it can commit to a differentiation. That is nothing but G naught. That is quiescent stage. Resting phase. It's also called as resting phase. It's a non-dividing phase, and this is how your nucleus looks in interphase, and then this is how it decondenses in prophase, and then forms a metaphase plate. And then it is pulled on the opposite sides in the anaphase, and then karyokinesis, and then cytokinesis. Next, which of these is not a constituent of animal cells? You all answered. It is cell wall. So, cell wall is nothing but a rigid structure which provides structural support, shape, and protection. Cell membrane is a semi-permeable membrane which helps in diffusion, osmolyte balance, and in intake and outtake of proteins or signals or ions that is required, and it separates the interior of the cell from the exterior environment. So this is how it is depicted diagrammatically, showing all the components of the cells more or less, and this is how the actual cell looks in a transmission electron microscope image. So this is some 5 lakh magnification of the cell. So even after such high magnification, you cannot see many of the components. You can only see the major components like Golgi, endoplasmic reticulum, nucleus, mitochondria, and uh, membranous structure and vacuoles. Next was in what phase the cell cycle DNA replicates. So that is S phase. So this is a picture I showed you all about the cell cycle chromophore, which is fluorescent in nature, and it is tagged to this PCNA protein, that is a proliferating cell nuclear antigen protein, which is active during the cell cycle synthesis division phase. So here you can see that in G2 phase how the cell looks when it is preparing for mitosis, and then when mitosis has to occur, the nucleus changes, it decondenses, it splits, and then it condenses again, and then after that the nucleus divides, and along with that the cell also divides, that is cytokinesis. After that the cell goes to first growth phase, that is the G1 phase, where it grows and becomes big, and now it is ready for replication of DNA. You can see the fluorescence here inside the nucleus, and you can see the replicating foci where the uh, newly synthesized DNA start to accumulate because they all start at different points. It's not like it starts at one point and it continues. It starts at different points in the DNA, and wherever it starts, the newly formed DNA starts to accumulate. And then this is the late S phase when most of the replication is complete. After this, it goes into growth phase again. That is the second growth phase. After this, again, growth phase to mitosis, similar cycle. Then next was RGD. So you have to know the alphabetical nomenclature for amino acids. So this is where this plays. So R is alphabet nomenclature for arginine, G is for glycine, D is for aspartic acid. So here they are asking what G stands for. G stands for glycine. So RGD is important because RGD is a very common and most important peptide motif which is involved in cell adhesion to the either to the extracellular matrix or to the adjacent cells. So this is used in various clinical applications for therapy, therapeutic targets, and for diagnosis. So in therapy, it is used to target tumor cells and attack them very specifically, and it leads to cell death. And then you can also give drugs conjugated to it to improve repair process where the cell has to attach now, and regenerative medicine, regenerative tissue has to attach. It speeds up the process of healing. So then in diagnosis, it is tagged with some radio labeled substance or some motif or some visually, contrasting agent, which will then go and bind to the cell, to the integrins, and then it will take up the tag, and the tag will then give you contrast while imaging. Next was dash implies that the specialized properties of the cell are lost irreversibly. Okay, so this is dedifferentiation. So once your differentiated cell goes back to a stem cell like state, it loses all its property. Now it can become anything that it wants. That is, it can de-differentiate. It can differentiate again back to the old type of cell, or it can re-differentiate into a newer type of cell, or it can, maintain its proliferative capacity and then enter into the cell cycle. Remember, it was differentiated was in G naught phase. So now from G naught, from quiescent phase, now it has to go back into that loop, that circle. G naught was outside the circle. It has to now go back into the circle. So then it will become a cell cycle, a cell capable of going into a cell cycle. If not, if none of these scenarios are completed, then the cell will be subjected to apoptosis or autophagy, and cell death will be programmed. Because this is a transient phase and the cell cannot stay in that phase. It has to do one or the other job. Then the other definitions for different terms here. Cell differentiation is the process by which a stem cell changes into a more specialized cell type. During this process, the cell loses its developmental potential and gains a specialized function and phenotype. Cell selection is a process that involves sorting of cells from a population with different properties, or its fate is determined. The cell is determined to become a particular type of cell. Now, cell adaptation. So, the capacity of the cell to answer different sorts of upgrades and negative ecological changes is adaptation. Whatever signals it gets, it will behave according to that signal, that is adaptation. If you remove that cell from that environment and then it has to now survive without all these signals, then it is de-adaptation. Then de-differentiation is a cellular process that causes a differentiated cell to convert back to a less differentiated state, sometimes even back to a stem cell. Stem cell is a very, uh, multipotent kind of cell which can now have the capacity to even give rise to many of the vital cell types. So dedifferentiation can be response to a stimulus and can be part of transdifferentiation. Transdifferentiation is nothing but when your dedifferentiation dedifferentiated cell is differentiated to some other type of cell. So that is transdifferentiation. And then where the cell refers to a less specialized state before creating new cells. That is also there where it enters into the cell cycle. Now, next is cardiac cells are mostly categorized as what type because they are heart cells, they are beating, they have to pump the blood, they contract, they expand, they are muscular in nature, they are muscle cells. This is the histology of muscle cells, both transverse section and longitudinal section. So we stain them to visualize them here also. This is called immunohistochemistry, where you use antibodies to see specific markers. You can do that also, specific proteins. This is eosin hematoxylin staining, where it stains the nucleus and the cytoplasm. So the blue here is the nucleus, the pink is the cytoplasm. So this is how different cells look after histology staining. So this is osteoblasts, osteoclasts, all these are bone cells. Then this is a neuron. Adjacent to that you can see tiny glial cells. You can see the cell body, the dendrites, the nucleus of the cell, the axon which is going behind, axon hillock, everything is visible. And then this is the epidermal cell histology where you can see the stratum kind of cellular arrangement and different layers of the skin. Next is HeLa cell line was derived from which cell line? So it is derived from cervical cancer cell lines. As you all answered, it is one of the very widely used cell lines for various experiments, various hypotheses, models, and for cancer treatment and for drug designing for cancer, etc. So here you can see the cells, how they look on a petri dish when you use a normal light microscope. And this is how they spread on the cells. You can see they are in a single layer. They are not overlapping on each other, which is very important. And here you can also see the concept of confluency, which is nothing but the percentage of the plate that is covered by the cells, where you can see a lower density or lower confluence of the cells in the left side image, which is around 60%, and then a higher confluency, which is more than 80 to 90%. And then you can get cells from ETCC, which are very certified cells, and then you can see the size of the cells. The scale bar given is 100 microns, so the cells are, you can see relatively how big they are. They are in micron length, less than 50 microns in size. And you can see spherical cells here. Few of them are dividing. This has divided just now. This is still dividing. Few of them are dying. So you can make out different cells in the microscope. So these are other cell lines of different type of cells, like muscle is L6, C2, C12. Then prostate cells are LAPC4, C42, 224, 22RV1. And then neuronal cancer cell lines are PC12 cell lines. So the next question is, which of these gases along with its buffer is used to maintain pH in the cell culture medium? That is carbon dioxide. So the buffer system that is generally used is a bicarbonate ion or a sodium bicarbonate salt. So now which gas will help this buffer to maintain the pH is CO2. So the CO2 is pumped, 5% CO2 generally is pumped into the incubator using big gas cylinders, and then the pumped CO2 will now get dissolved into the media. It will combine with the water, it will become carbonic acid. Carbonic acid will maintain the H plus ion balance and proton balance, and then it will also combine with the bicarbonate and become bicarbonate ion, which can be also stored as a reserve as bicarbonate. All these are stoichiometric reaction which has to be maintained in equilibrium. They are both reversible, they go both ways. So everything should be maintained in equilibrium. That is the main reason all this buffer and CO2 has been put in place. Along with this, I also spoke about the phenol red indicator, which helps us to detect the decrease in pH or increase in pH, either to acidic or basic. Mostly it becomes acidic because you grow cells in them and cells secrete uh, many substances which are acidic in nature, which makes the media acidic in nature. And along with pH check, this also serves as a contamination check when it goes to very bright yellow. Next is uh, what implies that the synthesis of specific products or other specialized functions are under regulatory control and can be reinduced if the right conditions are provided. This is differentiation. Like I said, implies that the synthesis of specific products or other specialized functions are under regulatory control, and now if reintroduced, in right conditions are provided, it can be reinduced to become a stem cell like cell. So this is differentiation. Then which of these is a non-adherent cell type? That is, what is a cell type which does not stick to the basement or whatever dish that you're growing it in. So in this, osteoblast is bone cells. They are very adherent. Hepatocytes are liver cells. They are also adherent. Neurons are also adherent. Erythrocytes are non-adherent because they are RBCs and they are always in blood, so they are in the suspended state. So when you grow them also, you have to grow in suspension media. So these are some of the common adherent and suspension cells. So the adherent cells are here fibroblast cells, which are, you can see, they look very similar to epithelial cells, but you can see they are more like longitudinal, and they have formation of fibers. So here these are MRC5 cells. These are vero epithelial cells. These are HUVEC endothelial cells. So endothelial cells are lining in the tubules and under the epithelial cells near capillaries, etc. So these are HUVEC endothelial cells. This is how they look like. And these are neuronal cells. This is how they look, NG108, and these are suspended cells. So suspended cells are mostly spherical in shape. Any cell, even if your adherent cell, you detach it from the media using trypsin or anything. Once you detach it and even they get suspended in the media, they also attain a spherical shape because that is where it can get the most volume. So all cells in suspended nature will be spherical. So you can see most of the primary T cells here. Okay, that's it for today. I hope everybody understood whatever I was trying to explain, and all your doubts are clarified.
Yes, ma'am.
Okay, if you have any more doubts, you are free to ask me. I will wait for 5 more minutes. If not, you can exit the class. Anyone with doubts? Okay, so I think I don't know what you're doing now. Okay, okay. I'm not gonna admit anybody because it's done. So let's wrap this class and then see you all next week. Bye, everybody. Thank you for attending the
Bye,
class.
ma'am. Thank you.
Thank you.